Christoph Sachsenmaier, Ribolution. Ribolution, using non-coding RNA for the next generation in diagnostics.
Ladies and gentlemen, I would like to convince you in the next 3 minutes that we are living in a post-genomic area of the Ribolution. And just to get to the brief history, about 15 years ago, when we sequenced the genome, we learned that over 95% of our genome was considered junk because it doesn't code for proteins. Now we know that the majority is transcribed into RNA, mostly into non-protein coding RNA. And we are just about to start to begin to understand the role of those non-coding RNAs in biology. We want to generate those non-coding RNAs into better next-generation diagnostics. The basis for that is that A, we now know that these are functionally involved in disease, and nature sends us conveniently packaged extracellular vesicles with RNA packaged in it so that we can use them as a liquid biopsy. We, that is a biomarker consortium under the roof of the Fraunhofer Society, applied this first to the case of prostate cancer. We have the current problem that we are using a very unspecific early detection test, the PSA test, to diagnose prostate cancer in men. If it is positive, the test, we need to undergo a very inconvenient and also partially dangerous biopsy. And just to find out that in 50 to 75% of the cases, there is no tumor. So we need a better diagnosis upfront of the biopsy to reduce unnecessary biopsy. To make 4 years of research short, what the colleagues at Fraunhofer found were about 40 novel non-coding RNA sequences that detect prostate cancer with a perfect 100% sensitivity and specificity. For those of you that are not in diagnostics, and so-called AUC of 1.0 means you hit the target every time. 0.5 means you can also roll the dice. PSA has about an AUC of about 0.6 in this field. So what we are going to plan, or what we are planning to do, is to develop non-invasive prostate cancer tests for different applications, first being a urine-based confirmatory test that should help us to reduce those unnecessary biopsies, but then move also into an early detection screening setting as well as a monitoring setting. And this brings me to the end of my presentation. Thank you.